Hello everybody, and in my video today we are going to be discussing in detail about M phase from cell cycle and cell division, class 11th botany Ncert. So we are going to be starting off with M phase. Okay, so what is M phase? Okay, you would have already studied about interphase, right? So we know certain things about interphase, and we have gap 1, synthesis, and gap 2. So these are the three types of interphase, and what actually happens on the interphase, what actually happens on the synthesis phase, this at all we are already clear. So now we are going to come to the M phase. Okay, so in this M phase also we have certain concepts in detail, and after studying about M phase we will also see about meiosis, mitosis. So in the M phase, first thing we need to understand is that this is the most dramatic period of the cell cycle. So M phase is the most dramatic period of the cell cycle. Why? Because all the components of the cell will be reorganized. Okay, so let us say like cooking something. Okay, so to cook something we need to prepare things, and that preparation is done during the interphase, and after the preparation the actual cooking process, that is the M phase. Okay, so everything will be reorganized. Let's say that all the food materials you have prepared and organized, you're gonna reorganize it on the M phase, like that we can say. Okay, so all the components of the cell will be reorganized. Okay, the number of chromosomes is the same, that we have already studied, isn't it? In the interphase we saw that the DNA will be replicated, will be doubled, but the chromosomes will always remain the same. So the number of chromosomes both in the parent cell and in the progeny is the same. It is also called as equational division. So why do we say it as equational division? This is very important. It is because the chromosome number is same in both the parent as well as the progeny cells. Though for convenience, mitosis has been divided into 4 stages of nuclear division. So previously we have seen about cytokinesis and everything. So now we are going to talk about karyokinesis. Karyokinesis means it is a nuclear division, so division of the nucleus. It is very essential to understand that cell division is a progressive process and very clear lines cannot be drawn. One thing we should remember when it comes to the cell division, okay, in this M phase, you cannot actually very clearly distinguish between every phase, because it is a progressive process. It's a sequence of events which very clearly you cannot depict the difference of every single stage. Okay, so we can say that M phase, it is associated with karyokinesis, and we are having prophase, metaphase, anaphase, and telophase. So these are nothing but M phase. Okay, the steps which occur in the M phase. First, let's see about prophase. Prophase is the first stage of karyokinesis of the mitosis. Okay, so all that we are talking about right now is mitosis. The interphase, the M phase, this is all mitosis, what we are seeing now. So what happens here is that it is the first stage. Okay, prophase is the first stage of the karyokinesis of the mitosis. So after the S and G2 phase, so you could have seen here, right? So here you might have observed that this whole part is M phase, right? And here gap 1, this is G1 phase, this is synthesis phase, and G2 phase. So here the whole M phase is there. So that's what they're saying. After the S and G2 phase, we find the M phase. In the S and G2 phase, the new DNA molecules are formed. Okay, so the prophase is marked by the initiation of condensation of chromosomal material. So let me just make it clear. Condensation of chromosomal material means it will be, first of all, it will be like this. It will be condensed. The chromosome material will be condensed. So later what happens is that, it will be tangled. So then condensation happens, which means it will be becoming untangled like this. Okay, so it is marked by the initiation of condensation. Condensation means this one, like untangled. Okay, this one is tangled and this one is untangled. Okay, the chromosomal material becomes untangled. Okay, that is called as condensation. During the process of condensation, the centrosome, okay, which had undergone duplication during the S phase of the interphase, now begins to move towards opposite pole. We have already discussed that during the S phase, okay, the centrosome will be duplicated, right? That one will move towards the opposite poles. So in the cell, if this is a cell, on this part and this part, on the opposite poles it will be moving. Okay, the completion of prophase, so they have given a summary of what all events will happen here. Okay, and one more thing we should remember is that at this time, okay, by the end of the prophase, when the materials they go up, okay, when the materials are up, like on the poles, okay, the centrosomes are moving towards the poles, right? During this time, no cell organelles. Like you cannot, like the cell cannot show nothing, like the cell cannot show anything like Golgi complex, endoplasmic reticulum, nucleolus, or even the nuclear envelope. It can show nothing during the end of the prophase. This is very important. So the chromosomal material condenses. Condenses means what? It untangles to form the chromosomes. They are seen to be composed of 2 chromatids. So 2 chromatids would be there, and they would be attached to one centromere. The centrosome, which had undergone duplication during the S phase, they will move towards the opposite poles, and each centrosome will have microtubules called asters. So what happens is that every centrosome will be having the asters. So asters are like the microtubules. The 2 asters together with the spindle fiber will form the mitotic apparatus. It is given here. Okay, this is the spindle fiber. So this is the mitotic apparatus. So this is the early prophase, which means, as you can see, the chromosomal material is tangled. Okay, but here it is separated. It is untangled now. Okay. So cells at this end of the prophase, when viewed under the microscope, they do not show any Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope. Now we move towards metaphase. So the complete disintegration of the nuclear envelope. Here, okay, somewhat it is not visible, but here completely it is not there. Completely the nuclear envelope is not there. It is the second phase, we know that. The chromosomes are separated through the cytoplasm of the cell. By this stage, condensation is completed. So here, completely the condensation is done, which means that completely it is untangled. And they can be observed clearly under the microscope also. And this then is a stage at which morphology of the chromosome is also easily studied. So at this stage you can also study about the chromosome, right? Because we can see it clearly under the microscope. It is easy to study the morphology of it. At this stage, the metaphase chromosome, it has 2 sister chromatids. Same, here also, prophase also 2 sister chromatids with 1 centromere. Yes, here also same only, and kinetochores are also observed. These structures serve as a site of attachment of spindle fiber. Now if you see, this is the metaphase. You are having the 2 chromatid, and this is a kinetochore. This thing that you see here is a kinetochore. So this is the spindle fiber, as I told you. It is attached to the kinetochore. Okay, so these structures serve as a site of attachment of spindle fibers to the chromosomes that are moved into position at the center of the cell. So the metaphase is characterized by all the chromosomes coming to lie at the equator. As you can see, they will be attaching and they would be coming to the equator. They will all come to the center, or we simply say they all come to the equator. So this structure you see is called as the metaphase plate. We will come to study about it. Coming to lie at the equator with 1 chromatid of each chromosome connected by its kinetochore to the spindle fiber. So 1 chromatid, okay, 1 chromatid. So this is 1 chromatid. Let's take this one part of the chromatid. Okay. So 1 chromatid is connected by its kinetochore, which means the kinetochore of 1 chromatid is connected to the spindle fiber from one pole. So this is one pole, this is another pole. So from one pole, the spindle fibers are connected to the kinetochore of this 1 chromatid, and also same in this way, this opposite side also. Okay, another sister chromatid means other chromatid. It is connected by its kinetochore. So this kinetochore of the sister chromatid is also connected to the spindle fiber of the opposite pole. So this arrangement that you see here is called as metaphase plate. Okay, this arrangement is called as metaphase plate. What arrangement? That you can see they all lie on the equator. Okay, connected to the spindle fibers from both the poles. So this is metaphase. This is a transition to metaphase. This is a complete the metaphase. Okay, so we have finally completed. They have given 2 key events here. Spindle fibers attached to kinetochores of the chromosome. Yeah, that's true. These are the spindle fibers attaching to the kinetochores. Then what's given? They have given that chromosomes are moved to spindle equator and they get aligned. So this is the metaphase plate. Okay, through the spindle fibers to both the poles. Finally, we are having anaphase. Okay, anaphase is the final, not the final phase. We still have telophase. Okay, at the onset of anaphase, each chromosome arranged at the metaphase is split simultaneously, and the 2 daughter chromatids. Now this is very important. You are having the metaphase plate, isn't it? From this metaphase plate, the 2 chromatids, okay, the 2 chromatids, which is now called as a daughter chromosome, begin the migration towards the opposite pole. So this will go there, okay, this will go there, and this would come here. Okay, as each chromosome moves away from the equator, from the equatorial plate, from this equator, this arrangement, they are moving opposite, right? The centromere mainly, it remains towards the pole, it directed towards the pole and hence at the leading edge, with the arms of the chromosome trailing behind. Thus, anaphase stage, what we say, what key events we say, centromeres are splitting. Okay, centromeres are going to split, and also the chromatids are going to be separated. Now the chromatids are going to move to the opposite pole. Okay, so this is the anaphase. Finally, they have moved towards the respective opposite pole. The final phase we observe here is a telophase. One thing we should remember is that telophase is exactly the opposite of anaphase. So telophase is exactly the opposite of anaphase. Why do I say that it is exactly the opposite means? Let us observe. What are the key events we saw? Prophase. Okay, exactly opposite to the prophase. So what are the key events we have discussed on the prophase? If you recall clearly, you will understand that no cell organelle is found. First thing is that no cell organelle would be found. Another thing, the chromosomal material will be tangled. So tangled means it will be like this, right? Be like this. So here what happens is that it will be untangled later. Okay, so the tangled chromosome will be untangled, right, in the prophase. Here what happens, again it decondenses, which means again it tangles. Okay, again it tangles. That's what it's in the telophase. At the beginning of the final stage of the karyokinesis. Final stage of karyokinesis is what? Final stage, right? That is a telophase. What happens is that the chromosomes that have reached the respective poles, again they decondense. They lose their individuality again, which means that if they are like this, how will the individuality be, right? So they can no longer be seen, and each of the chromatin material tends to collect at two poles. So here again we can find the chromatin material at the two poles. Okay. What are the key events? Chromosomes cluster at the opposite spindle poles, and the identity is also lost. Plus we see that the nuclear envelope again comes. Okay, in the first time in the prophase we saw that there was no cell organelle, or there was no nuclear envelope, no Golgi complex, no nucleolus, nothing was there. But here again it develops. Nuclear envelope is going to be developing around the chromosome. Nucleolus, Golgi complex, and the endoplasmic reticulum reform. Okay, so this is it about the telophase. Then we are having cytokinesis. Then we will talk about significance of mitosis, and then we will end this video. So later we will discuss in brief about meiosis. As of cytokinesis, we know that what we have discussed till now is karyokinesis, which means a division of the nucleus. So the M phase, the interphase, everything we spoke about karyokinesis. Now let us talk about cytokinesis. Cytokinesis, similar to karyokinesis, talks about cytoplasmic division. Cytoplasmic division. So mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, but the cell itself is divided into two daughter cells, right? So if karyokinesis is there, obviously the cytokinesis should be there. Otherwise what happens? The multinucleated condition will be seen, okay, which means that the nucleus tends on be dividing and many nucleus will be formed. At one stage when the nucleus is divided, even the cytoplasm should divide. Only then two daughter individual can be formed, isn't it? The same similar way they are saying here. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. So in a plasma membrane, a furrow, the appearance of a furrow they are saying. The furrow will deepen and ultimately joins the center. So what happens is that in the plasma membrane, we are talking about animal cell. So let us focus on it now. We are speaking of which? We are speaking of animal cell. So let us focus on it. So in animal cell, what happens? A furrow is found in the plasma membrane. Where it is found? In the plasma membrane. This furrow will join the center. Okay. So furrow means the periphery. From the periphery, they'll join the center. Okay, this happens. So dividing the cytoplasm into two. In the case of plant, now let us come to plant. What we have seen in the animal, from the periphery to center. In the plant, what happens? In the plant, center to periphery. Okay, they are enclosed by relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism, because in animal, animal and plant cell, the difference is cell wall, right? So that's why we say like that. So here what happens is that they undergo cytokinesis by different mechanism compared to animals. In plant cells, the wall formation starts in the center. Okay, then it grows outwards. See, center to outwards, center to outwards. Okay, in animal it is outwards to center. In the plants it is center to outwards. New cell wall will be formed up. Begins with the formation of a new cell wall, that is called a cell plate. Cell plate means it is nothing but the lamella. Okay, at the time of cytoplasmic division, organelles like mitochondria, everything will be distributed equally between the two daughter cell. So that's what I already told, right? If karyokinesis is not followed by cytokinesis, that means only the nuclear division is going to happen and the cytoplasmic division is not going to happen. In such case, what will happen means multinucleate condition we find, which is called as syncytium, or we say cenocytic. Okay, for that they have given example, liquid endosperm in coconut, which is nothing but the coconut water. In case the cytoplasmic division is not going to happen, keep on the karyokinesis is going to happen, then we say it as a multinucleate condition. Finally, talking of significance of mitosis, mostly, okay, this is easy part only, like nothing much difficult. So mitosis is usually restricted to diploid cell. Okay, diploid cells means 2 end cells. Okay, generally in the case of diploid cells only we will be finding the mitosis. However, in some lower plants even the haploid cells also can be used, or some social insects like we have discussed already, right? Haploid cells means N, okay, like honeybees at all. Okay, we have already seen in the plants and honeybees at all, right? So they also, haploid cells also will divide. It is very essential to understand the significance of the divisions. Why is it important? Mitosis results in the production of daughter cells, diploid daughter cells, first thing. Secondary, the growth of an organism, be it any organism, the growth is because of mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleus-cytoplasmic ratio. That is, if cytoplasm is not going to be divided, then keep on the multinucleate condition will be observed. So it is very essential to maintain the same ratio, how nuclear and cytoplasmic ratio. That ratio should be very importantly maintained. A very significant contribution of mitosis is cell repair. It also helps in cell repair. So, you know what happens is that cell repair means during injury and wound at all, that particular tissue or the cell would be restored. Like in the case of blood cells at all, the cells lining the gut or the upper layer of epidermis at all, the restoring it will be replaced. Okay, and mitotic divisions in the plants are found on the meristematic tissue. In animal you can find in these places. In the plants you can find on the meristematic tissues. Okay, that is the apical and the lateral cambium result in continuous growth of plants. Okay, so that's it about today's video. We have discussed about the M phase, we spoke about the cytokinesis, we spoke about the significance of mitosis. Finally, we are having meiosis pending, which we will see on the next video. Thank you for watching till the end. If you like my video, please subscribe to my channel and also hit the like button. Thank you. Bye-bye.